drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. All right, Professor Michael Snyder, thank you for joining me.
Pleasure to be here.
Can you start off by just telling everyone a little bit about who you are and what your lab studies?
Sure. I'm a professor of genetics at Stanford University School of Medicine. I direct the Center for Genomics and Personalized Medicine. Our specialty is big data. Back at a time, people used to study genes one at a time, and we came up with ways of studying them all at once. It used to be on yeast. And so I think that's kind of guided our research ever since, trying to look at problems in a more comprehensive sense.
Mm-hmm. So can you give us a sense for what is the breadth of questions your lab looks at, and what are some of just give us a basic sense of some of the tools and technologies you deploy?
Sure. Well, I think what drives us is that we think medicine is broken. That is, we practice sick care, not health care. And so we're trying to transform that in many different ways. As an example, when we go to a doctor's office, we typically measure very few things there, and some of it's of questionable value. And then we treat people based on population averages. What we're trying to do is get a much, much more complete picture of people's health, do a lot of it by monitoring at home, and then actually treat people based on their personal profiles and their trajectories, which may or may not be like a population average. A good example is you've probably been told since you're little, your temperature when you put a thermometer in your mouth is 90.6. But if you actually look at the data that are out there, it's more like 97.7, according to a big study out of Stanford.
On average.
People, on average. But the more important point is there's a spread. There's another study that says 25th quartile is 94.6, the 75th quartile is 99.1.
Wow. So from 94 and a half all the way up to 99 is where most people sit.
Yeah, that's correct. And so what that means, if your normal healthy baseline is 94.6, you go to a physician today, they measure at 98.6, they're going to tell you you're healthy, you're normal, everything's fine. But I guarantee if you're up 4 degrees over your baseline, you're not healthy. Something's up.
Right. And I suppose another thing there is that number, that temperature is itself dynamic. It's going to be a function of, what you eat, for example.
It is to some extent, yeah. And you'll have a little bit of fluctuation on a circadian pattern. Yep.
Okay. And then, so what kind of, just give us a basic sense before we go into some detail on some of it, because I know that you do a lot. What sort of technologies are you using? You're more of a molecular lab. You get down to the molecular level.
Yeah, molecular and genetics. Although these days we're getting a lot into wearables and physiology. So basically, I guess what we did about 15 years ago, stemming off some work we used to do on studying biological systems, is we tried deep profiling on people with the idea of monitoring health. So the technologies we use will sequence people's genome to try and make predictions about their genetic risk. We'll draw blood and urine and stool to look at their microbiome, and we'll do very deep measurements based on RNA, their proteins, their metabolites and such. We've invented some of those technologies that are used today. Something called RNA-seq is one of these methods that came out of our lab. It's a way of looking at all your RNA molecules. Anyway, we'll measure that out of blood. Some of like metabolomics and things, your metabolites, will measure that out of urine. Your microbiome, you may or may not know, it's super important for your health. It makes lots of essential vitamins, breaks down your food. We'll measure that as well. And so one aspect is to take very, very deep data on people, and the other aspect is to do it over time. So we'll actually profile people while they're healthy, not while they're sick, see what a healthy profile looks like. We do it every 3 months and additional times if they get ill, and we'll profile them over time. And the idea is to see what is your healthy profile, how's it changing, what's the difference between, say, you and me. How are we the same? How are we different? Turns out we're quite different. And in fact, this is why what we're doing we think is so important for health monitoring. Turns out that a sick you is still closer to a healthy you than it is to me. Meaning, sorry, the difference between people is greater than the difference between a healthy and a sick profile.
So if we did just some of the profiling you're talking about, never mind what the details are at this moment. You looked at my profile and your profile, and then you looked at my profile again after, say, I got sick. My healthy profile and my sick profile would probably be closer to each other than my healthy profile would be to your healthy profile.
Correct. Because we're so different from one another. And that's why it's important to know your healthy profile. So when you get ill, you can detect that shift.
And I guess kind of what you're getting at, or what some of the subtext is here based on what you said initially, is when the way that we do medicine today, in essence, people are only going in and getting a partial sick profile after something goes wrong.
Correct. And so it's crazy. And then you try and figure out what's going on. Whereas if you're following them, again, while they're healthy, you can see what shifts, you have a better idea what's going on. Not perfect, but we take literally 135,000 measurements every time we measure someone. So we're getting a much, much deeper profile. The analogy we like to use is that if your health is a 1,000-piece jigsaw puzzle, we're trying to get 600 or 700 pieces versus, say, 5 or 6, we would argue you get today.
Yeah.
You have very incomplete pictures.
Yeah. And not only is it 5 or 6, but there's a pretty good chance that one or more of those mere 5 or 6 pieces aren't even relevant to what's going on.
Yeah, your heart rate in a doctor's office for most people is worthless.
Why is that?
Because they get nervous.
Yeah.
Heart rate jumps up 20 points.
Yeah.
Whereas, believe it or not, if I were to pull your heart rate off of a smart watch, which is we do a lot with wearables as well, first thing in the morning, it's a pretty good indicator what your baseline heart rate is. And if you're stressed, either mentally or, say, physically, like you're coming down with something, your heart rate will jump up, and we can pick that up.
So there's a lot of directions we can go with this. I'm going to start us on one track, and we'll just kind of see where we end up with this conversation.
Sure.
But obviously, something that's top of mind for a lot of people is diet and how diet affects metabolic health. Many different aspects of that we could discuss. What are some of the major questions in your lab that you've been addressing recently that have to do with the connection between diet, what you're eating, and what your metabolic health looks like?
Yeah, we're doing a lot in the area of glucose control, believe it or not, and diabetes and prediabetes more than that. So because, again, we're pretty keen on keeping people healthy. So this idea of prevention is, which is not so well exercised in today's healthcare system. Again, we do sick care. So it turns out, well, first of all, I'm diabetic. You may not know it if you were to see me in person. I'm a pretty skinny guy. Yeah.
Type 1 or type 2?
A weird type 2.
Really?
So that's another thing. Diabetes is not just type 1 and type 2. It's probably more like type 1 through type 50.
Wow.
So type 2, we know we can subtype. For example, some people don't make insulin well. Other people don't release insulin from the pancreas. That's me, actually. Most people, their cells don't respond to insulin.
I see.
Insulin lets your cells take up glucose. That's the most common form of diabetes, but there are many subtypes.
Yeah. So hold on. I just want to make sure I understand, because I actually didn't know that there was that much difference there. So type 2, broadly speaking, that means you have acquired insulin resistance and become diabetic late in life, as opposed to being getting it early in life because your pancreatic beta cells got destroyed. So
correct. So that's type 1, by the way.
Yeah, that's type 1.
Subscribe. Type 1.
2, most commonly, people are producing insulin, but their cells are no longer responding to insulin as much. But
Right.
you have a different version.
That's correct. In fact, and it's not unheard of either. It's not common. So my version is that I do make insulin. My cells respond, but I don't release insulin from the pancreas.
Hmm.
And not only that, it turns out I don't respond to metformin, which is also kind of unusual.
And that would be a common drug that people do respond to.
Yep, and that's what most people do, and it works pretty well for most type 2 diabetics, and it's generally viewed as quite safe. These days, there's a whole bunch of other drugs out there you may know. These Ozempic derivatives, they mimic a hormone called incretin, and they're extremely effective. So anyway, I didn't respond to the most common drug. It's metformin, but I did respond to something that stimulates insulin release from the pancreas. It's called rapinolide. And I actually worked well with that. Now, these days, I'm on actually one of these GLPs because they work even better for me. But yeah, so these Ozempic kinds of drugs. I happen to be on one called Mounjaro.
Yeah.
Anyway, they work incredibly well on me, but we think this is really important because diabetes has many subtypes, and the way you treat people very much depends on the subtype you are. That is like I was just describing. I respond to certain drugs, but not others. The other thing we're discovering, here's an area of research that's very active for us, is we all respond to foods differently from our glucose response. That is to say, some people, so first of all, backing up a little bit, here's the problem. 11.9% of people in the US are diabetic, 33% are pre-diabetic, and they're going to go on and become diabetic in their lifetime, the vast majority of those.
So roughly speaking, something like half-ish of adults are either diabetic or pre-diabetic in some way.
Right. It's totally out of control, and people don't take it as seriously as it should. This is an endemic that's quite serious because it leads to, heart failure, all these problems, increased cancer later in life.
We often think of epidemics in the context of infectious disease, but we have an epidemic of this metabolic disorder.
We really do. Obesity and diabetes, and they're not identical, but they do go hand in hand. So most people who are diabetic are overweight, but not all. There's a number of people like me who are thin diabetics, probably because there's some subtypes different. So anyway, we're trying to figure out what the subtypes are and then how to better manage it. First through lifestyle, because we think that's probably a healthier way to do this. And one form of lifestyle is what you eat. I'm really a firm believer food is medicine. And I think more and more we're seeing this.
Yeah.
You may not realize this, but people eat 40,000 times more sugar now than they did at the end of World War II.
40,000.
40,000.
How do we know that? What's the basic
you just looked at how much diet there wasn't as much sugar around now. Now sugar is in everything.
When you say sugar, do you mean sort of the number of glucose molecules that end up in their body, or do you mean the sort of the mass of added sugars?
Added sugars.
Okay.
Incredible. Yeah. It's true that people that had carbs, but it's a healthier form. So ultra processed foods, a good example, they're usually loaded with lots of sugar. And if you look at how much sugar is in a bottle of ketchup, it's most of the bottle. It's incredible.
How do we get to that? Hold on. 40,000 times?
40,000.
But I'm a little perplexed here. So no one's eating 40,000 grams of added sugar per day, but people, 100 years ago were probably eating one or two.
Maybe not.
Really?
Straight up sugar, yeah.
Yeah.
Right at the end of World War II, probably not as much. Yeah, and just look at people's shape. If you look at diagrams of crowds, or not the photographs of crowds from people in the 1940s, they don't look at all like they look now.
So in essence, what you're saying is, before World War II, there was little to no added sugars in things, but today it's kind of in everything.
It's in everything.
Yeah.
And, kids, they get on sugar right from the get-go. Mine did.
Yeah, from in the womb even.
Yeah. Yeah, that's true too. So anyway, we have a problem. And so one is simply getting rid of that. Now, I did that initially. It's how I got, when I first became diabetic. I'm a very interesting case, by the way. I'm genetically, I was predicted from my genome. My DNA sequence predicted my diabetes. And then, but I wasn't diabetic at the time. I only became diabetic after a very nasty viral infection. It's called respiratory syncytial virus.
When was this in your life? How old were you?
I was, it would have been 2011, so I would have been, that's 14 years ago. So I guess I was 54.
Wow. So middle-aged, and this was you had a genetic predisposition of some kind, but you needed this sort of environmental trigger for this disorder to manifest.
Correct. Yep. Yeah, and scientifically, it's very interesting. Obviously, I wish it hadn't happened, but scientifically, it turns out if you've heard of epigenetics, where you can get modifications on your DNA,
Mhm.
it turns out my metabolic genes got modified, and that's when my sugar went off. If you look at the kinetics of all this, meaning we've done very detailed analysis of my epigenetics. And you can see my sugar has gone off twice. When I first became diabetic, my methylation pattern shifted. It took about a year. I got it under control through lifestyle changes. I cut out all exogenous sugars. Yeah, and I was doing a lot of running and got my sugar levels down to normal. And then it actually came back after I stopped running. And I also got a
Huh.
second viral infection. So it's not 100% clear. I started running again, but I never got all the way down. I got it partially under control. But each time my glucose went off, my epigenetics change over a bunch of metabolic genes. It's
I
quite
see.
fascinating
So,
scientifically.
yeah. So in essence, it sounds like what happened is you got an infection. It sounded like a respiratory viral infection.
Yeah, that's right.
And then obviously your body reacts to this. There's an immune reaction. There's some kind of inflammation. There's stuff happening in your body. As a side effect of that, you had changes in DNA methylation. So different genes got turned on and turned off.
Right.
Some of those genes had to do with glucose regulation.
Yep, that's exactly right. And it's interesting now because, and that was the first time that had been shown where a viral infection, it's been suggested for type 1 previously, but not for type 2. And again, I'm a weird type 2. But now you may or may not realize that with COVID, somewhere between 2 to 4% of people become diabetic after a
Really?
COVID infection. Yeah, so it's probably a more general phenomenon than you think.
Yeah.
Quite a few people are getting something called chronic fatigue syndrome, if you've ever heard
Yes.
of
I've
that.
been reading about this.
Yeah, it turns out it's a lot more prevalent than people realize.
Right.
That's also often thought to be associated with the viral infection or some
Yeah.
other bacterial
Well, yeah.
infection.
Well,
Some
did
other,
you see the new Yale study that just came out?
I did not, no.
So,
Oh, the one from Akio, yeah,
yes.
yes,
Yes.
yeah.
Basically there's a post-vaccination syndrome that you can see. And even, we're not going to dive into this, I don't think, but the punchline here is they were able to segment people based on whether or not they had 0 to 4 doses of one of the COVID vaccines, and also determine whether or not they ever had a previous SARS-CoV-2 infection, including whether, even if they were asymptomatic and didn't know it. And so, you had people that had a vaccine, that didn't have a vaccine, that had prior infection, that didn't have prior infection, four
Uh-huh.
different cohorts there. They were able to show that people who had multiple doses of the vaccine but no prior infection, some of those people even had this post-vaccination syndrome that was probably
Mhm.
coming from the spike protein.
I see, but probably lower frequency than those who didn't get it. Is that right? Do you know?
I don't know about that. You saw it in both
We'll
groups.
have to
Yeah.
look it up. Yeah, we'll both
Yeah.
go take a
There
look
was there was a small number of people who had spike protein in their blood almost 2 years later who had never had a prior infection.
That's showing up now more and more commonly. Yeah, this idea that this thing is lingering in some fashion.
Yeah.
And it's that in general for people who get long COVID, if they haven't been vaccinated, the probability of getting long COVID goes up. So vaccination does, in general, seem to help protect.
Yeah. But I think what
It's
you're
not
saying
to say
is,
sometimes there aren't complications. There are.
well, yeah, I think the picture that seems to be emerging in my mind is so there's an interaction at two levels, at least. One, with the vaccines aren't as good as we would have hoped at preventing infection, but they do seem to prevent severe COVID, which would imply a lower overall spike protein load from an infection if you've gotten a vaccine. But at the same time, the mRNA vaccines, are engineered to have highly stabilized mRNAs that will produce a lot of spike protein potentially. And we're talking about people now who've gotten 2, 3, 4, even 5 or 6 doses of this, and there's probably something going on with the number of total exposures to spike, either from infection or from a vaccination that people have had.
Yeah. Okay. Anyway, it's pretty clear. Yeah, people who get COVID, it does linger longer in some cases. Yeah.
So anyways,
Yeah.
going back to your case, Can you talk a little bit about like how, when you first became diabetic, you've got this less common version of type 2 diabetes. How did, as you discovered this and as you started to get talking to your doctor about it, getting treated for it, how did that play out? I would imagine that maybe your physician probably wanted to start by treating you the way you would treat, the average type 2 diabetic, but then in your case, this probably wasn't the right thing to do.
Yeah, that's definitely the case. I mean, in my case, I caught it right away.
Mhm.
Now, I'm not saying everybody gets triggered that way.
Yeah.
We're studying this group of people, and I'd say most people just gradually get diabetic. But there's at least another person in our cohort who was like me, who spiked their way there as though something triggered it. So I think either way could happen. In my case, I just quickly changed my lifestyle because I saw it happening right away, and I gradually, as I say, got it under control, and that worked. And so I was pretty happy with that.
Mhm. How did you notice it was happening? What was
Well, because I profile myself a lot.
Okay, so
I'm one of my guinea pigs. So
I see.
we're studying this group of 110-ish people. We've been doing it now for well, for me, 15 years, and for this group, it's been about 12 years. We've been profiling them very, very deeply, and again, with the goal of trying to understand. It was mostly just, can we use big data to better manage people's health? Can we use genome sequencing, for example, to better predict whether we can, to see if we can, again, manage your health? And in some cases, for example, from the genome sequencing, we found someone who had a heart problem. He didn't know but his genome said something was off. Or he should have this heart defect, I should say. And it turns out he did. He, and fairly young, didn't know it, and so he's on medication now. It was, again, another person was at high risk for cancer. They did a whole body MRI, and it turns out they actually had early thyroid cancer. So we're kind of bringing these technologies in to see if we can, again, better manage people's health. And in the case of genome sequencing, predict what they're at risk for, and then keep an eye out for those things. In the case of these deep data monitoring from blood, see if we see something off. We caught someone with early lymphoma, for example, by the steep profiling, and 2 people with precancers. So we think this deep data really does give a better picture of your health and lets you catch things before symptoms. These people did not have symptoms. And so that's the key. So back to my diabetes. I know I'm digressing a bit here, I guess. Anyway, when we ultimately, I couldn't quite get it under control just by lifestyle. I was changing my diet. I'd cut out all the sugars. I don't eat any of that stuff. I was being pretty active, meaning I was running about 45 minutes a run, say 4 times a week. It's about what my schedule could accommodate. And yet my glucose just gradually kept creeping up and up and up. So then I finally bit the bullet. I did try metformin, which is the thing you would normally take, and I didn't respond. I increased the dose. I still didn't respond. And then, believe it or not, I tried. So I hadn't figured out what my defect was yet. But I started lifting weights, believe it or not, because the idea was that increasing your muscle mass is better for glucose homeostasis. And it worked in the sense I gained 10 pounds of muscle mass.
Wow.
But it didn't help my glucose at all.
Really?
Do a whole body MRI all the time too, so I could actually quantify all this, my muscle mass increase. And it didn't help the glucose. And then it just kept creeping up and up and up. And then finally, through more measurements, I realized what was going on, that I was not releasing. I knew I was insulin sensitive, and then I realized I wasn't releasing it from the pancreas.
So you had non-insulin resistant type 2 diabetes. Your blood sugar is going up too much because you're not producing enough insulin.
Right. Well, I'm producing it. I'm just not releasing it from the pancreas.
Oh, I see. Okay, so the cells make it, but they're not releasing it.
It sounds like a subtle difference, but it makes a big difference from the drug standpoint, meaning rapinolide is known to promote release from the pancreas. And that works.
You don't need something that cranks up production because you already have production.
Correct. Yeah. So it's interesting. And so this is why we think subtyping diabetes is a big deal, which is not done at all now. If we can do that. And it turns out, here's some studies we're doing now that's a lot of fun. Different people will spike their glucose to different foods. So they have these things called continuous glucose monitors. You may
Yep.
be familiar with those. So they measure your glucose every 5 minutes. And it turns out that our claim to fame, so to speak, is we started putting these on so-called normal people and pre-diabetics and discovered many of them have very bad glucose control. So obviously diabetics spike like mad after they eat something sugary. Turns out a lot of normal people do it, and many pre-diabetics spike pretty badly too. Some just as bad as diabetics, and they don't know it.
Hmm.
So, and it turns out different people spike to different foods. So some people, their glucose will spike to banana, others to pasta, some to white bread, some to brown bread, some to potatoes. We're all different. And Aaron Siegel, a different group, has shown that about 21% of that is due to your microbiome, the microbes in your gut, and how presumably how they metabolize fiber and sugar and such. But most of it's not explained. What we found is that for if you're insulin resistant, you'll spike to potatoes and pasta. So different foods have different response depending on your subtype.
Huh.
So we're trying to break this
I see.
down even further.
Is that a pretty robust finding that if you're insulin resistant, you will spike to those starchy foods?
Yes.
Okay.
It is. We all spike to white rice, by the way. White rice is very spiky. Yeah.
Okay.
And yeah, but so we think it's probably, this is just hypothesis, but we think it's the resistant starch in there that might be responsible. Now, how that interplays with the insulin resistance, we have some ideas, but it's not fully clear yet.
Yeah, yeah.
So anyway, we ultimately want to, basically the end result out of all this is we want to build personalized diet programs.
Yep.
So if you know what spikes you, you avoid that and you eat the things that don't.
Right.
Okay. The idea is to push off then getting prediabetes and ideally diabetes for as long as possible. I don't know if you'll push it off forever, but you can try.
So how often are you getting profiling?
Constantly. So I wear these monitors. So normally two of my watches broke, but you can say I have, well, here's a ring and two watches. Normally I have four. My hearing aids are sensors as well.
Yeah.
So at one level, I'm measuring my physiology all the time. At another level, I do these blood profiles and I wear a glucose monitor quite often. I'm not wearing one right now, but I do these omics profiling probably every 2 months or so. And if an adverse event like a viral infection, I take more samplings.
Yeah.
So I do a lot of this.
Is there any so all this stuff is very cool. I've done some of this to some extent myself. One thing that we need, and maybe if I'm lucky, you'll tell me we have it to some extent. Is there any platform that can integrate the data from all of these devices so you can have it all lined up in one place?
Yeah, I should have started this program by saying I'm 100% conflicted because we spin off companies and what we're doing is academic. We're trying to use these technologies to see what works, what's useful, what we can learn from them. But when things actually make sense, we'll spin this off as a company. So as an example, we have a continuous glucose monitoring company. It's called January Eye, and they build these personalized programs. They have a 50-some million food database. It's huge, and they can help you with all these predictions. Of course, they use AI for all this. So where I'm going with this, we have another company. It's a big data company. It's called QBio. They do whole body MRI and do a medical version of what I just said. They won't do the microbiome because we don't really know how to act on that yet, but they'll make all kinds of measurements. And right away, they've discovered, and they'll pull the data in and display it in a fashion that is so logical. To your point about integration, they can bring in the MRI data and the molecular measurements and the genetics and display it in a way that says, all right, oh, look, these three things are off in your cardiovascular space. Keep an eye on that. Versus if you get your medical report today, they put the ions and things all together that don't necessarily make biological sense, right? You can't read it easily. You have to flip through the pages and try and figure it out for yourself. It's crazy. So basically, it's a matter of organizing the data in a very useful fashion. These days, of course, you can use AI to pull that information together and give specific recommendations. So we haven't talked about this yet, but we've set up another method for doing home microsampling and doing deep profiles on people, meaning you take little drops of blood. We used to do it from the fingertip. Now we do it from the shoulder. You mail it in, and we can measure 7,000 molecules from that drop of blood. And now I know what that sounds like, but ours actually works.
Yeah. No, for those listening, the technology has progressed since the infamous days of Elizabeth Holmes, and you can actually do blood work on relatively small amounts of blood and get lots of measurements done. I've done this myself.
Yeah. Yeah. So anyway, we spun off a company there called Iolo that will measure 650 metabolites, pretty reproducibly.
What's that one called?
It's called Iolo, IOLLO. And and so you give the blood, you mail it in, and they give you back a report on these 20 wellness categories. Like they'll measure your oxidative stress, your inflammation, heart, and various things, and they even measure your biological age. Now, I know there's a lot of aging clocks out there.
Yep.
And so I think they're pretty good. Like the True Diagnostics methylation clock is actually the from Steve Horvath originally. I think it's pretty good. But what's special about the metabolomics clock is it's actionable. And same with this information I was telling you. So they'll break down, we call it ageotypes, your aging patterns. And so we can see your heart age and your kidney age and oxidative stress and these various things. And so, and then you can use AI to basically take the information you give them about yourself, plus what's in the literature, and they make very specific recommendations. So it's not just exercise more, eat better. It's really like eat this exactly. You've been eating this already. We'll try this kind of thing. And so it works actually in the sense that people do improve their markers after they get the report back. And the hope, of course, is that will lead to longer, healthier lives. That's the goal anyway.
Yeah. I want to talk about some recent work that you've done. And I guess what I want people to take away from this section is to have a better grasp on how to connect the dots all the way from what you eat to what's being produced in your body or by your microbiome to a change in metabolism via changes in gene expression. So linking, things like dietary fiber or other dietary components to your microbiome, to gene expression in your body and epigenetics, to metabolic change at the end. So you recently did a study that looked at something called short chain fatty acids and how these affect gene expression, how they're produced in the body. Let's start out really basic here. What are short chain fatty acids, and what are some of the major ones that people might hear about?
Yeah, they're these little molecules, acetate, propionate, and butyrate are probably the three most common ones. And so it turns out that they've been known for a long time to be healthy for you. They, boost your immune system, and exactly how they're healthy isn't fully understood, and certainly how they work is not understood. But it's known that everybody knows fiber is good for you. And we have a number of studies here, back to your earlier question about nutrition. We're actually trying to break this down because there are many different kinds of fibers. There are long chain, short chain, hydrophobic, hydrophilic, positive, negative. It's all over the map. Mm-hmm. And so we've been testing different fibers.
And I guess with the unifying theme here for like what counts as fiber, it's something that we cannot digest on our own, and therefore microbes inside of us must break down for us.
Bingo. Yes. Yeah, absolutely right. And so, and the fibers themselves, I mean, some of them have these compounds called polyphenols. You may have heard of them. They're getting more and more attention. They're antioxidants and probably have a lot of other health benefits too that people don't fully, know yet. But anyway, your microbiome breaks this down, and certain microbes then will actually produce short chain fatty acids as a consequence of all this. And so these short chain fatty acids are generally thought to be quite good for you. And there's plenty of evidence for that. So what people had been generally thinking is that there's an important modification on your histones, of things that package your DNA and affect gene expression. And basically, the prevailing view was that there's a very important modification. It's called acetylation. And these short chain fatty acids that mostly get produced by the fibers, they're called propionate and butyrate. They were thought to actually affect this other modification called acetylation.
Hmm.
And I think what was kind of nice, what we did, and it stems from another lab's work, they showed that these things can actually show up on your chromatin itself, the packaging of your DNA. And I know this is getting pretty technical, but what we did show was that we actually went in and showed, yes, they're really showing up there. We actually saw exactly where they're sitting on your colon cells from, cells grown in a test tube in a petri dish. We could actually see exactly what genes were getting modified, if you will. Their packaging was getting modified by these short chain fatty acids. So the idea is that we can make a direct link from fiber
Yeah.
to the actual genes that are getting affected. And it turns out it's genes important for gut integrity. They're these connections between your gut cells. They're called gap junctions and such. And they're regulated by
Yeah.
these fiber modifications, these short chain fatty acids.
I see. So if I can summarize what you've just said so far, long story short, your gut needs to have high integrity, meaning that the physical connections between the cells of the gut need to be in a good state. Let's just say that you don't want them too loose because then you'll have a leaky gut. Say you probably don't want them too tight anyway. You want them to be healthy, whatever exactly that means. That healthiness depends in part on the proteins they're making, which of course depends in which genes are being turned on and turned off and to what extent. What you just told us is that dietary fiber, you can eat fiber. Microbes, if they're in your gut, the right ones, will turn that fiber into short chain fatty acids. And these short chain fatty acids actually go in and affect gene expression. And some of the genes they affect are the ones that control the physical integrity of the gut.
You nailed it, and I appreciate you translating it for me. No,
So,
you're spot on. Yeah. So the bottom line is then fiber is making key molecules that are important for gut integrity. And as you point out, if you don't have that, you get a leaky gut, and then your immune system starts interacting. The microbes can get inside your bloodstream in some cases if you have a leaky gut. It's a big deal.
yeah.
So you really want a great intestinal. Both
Yep.
mucosal layer and having the cells, joined together.
So one question I want to ask you about this, and this is something I've been reading about, and my thinking is, it has changed. I've now, my thinking has changed. I'll just leave it at that. Something I often hear, and I want to get you to very carefully parse this for me. So it's unquestionably true that dietary fiber can get metabolized into short chain fatty acids that lead to good stuff for your health via the gut microbes that are in our gut. The question I have is that the only possible way to make these short chain fatty acids? And just to put all my cards on the table, it appears the answer to that is actually no. It's not the only way to get these things. What can you tell us about that?
Well, you can eat some of these things, believe it or not. You can take these as a supplement. I don't know this area so well myself, so it's not one I can probably talk about with any, informed state here. But
one way to get these things is through fiber.
For sure.
Yeah.
And I think people like the concept of fiber. I certainly do, because you do need again, for the mucosal layer of your intestine and just keeping that microbiome healthy. You may or may not know that 70% of your immune cells are in your gut.
Mm-hmm.
And a lot of it's there to keep that balance between your immune system and the microbiome that's there, keep everything in check. And so that's a big deal. And you really want a healthy balance there. And so, yeah, I think fiber is a great way to get it. As I mentioned before, it has certain fibers have these polyphenols, which are antioxidants, amongst other things, that are very valuable for your health as well. And so, yeah, your fiber is fiber is basically setting up a fermentation chamber in your colon that's just very, very valuable for your health. And so, sure, you could probably get some of this short chain fatty acid through diet, but it's not going to be quite the same.
Yeah, the work that I'm thinking of here, and there's I think very little of this work has actually been done, but I think his name is Peter Turnbaugh at UCSF, I believe. So he did a small trial in humans a number of years ago. They did a plant-based diet with fiber. They did an animal-based diet, which was basically just meat, eggs, and cheese. It had essentially little to no fiber. It was only about 5% carbohydrate. And what they showed is that in that animal-based diet, even though it lacked fiber, they nonetheless had butyrate and acetate production. It was somewhat lower than with the plant-based diet, but it was there. But then you had this increase in these other short chain fatty acids, isobutyrate, and some other ones. And
Right.
so it showed that, at least in principle, for some people, you can get production of you have a different profile of short chain fatty acids, but you still get them even in the absence of dietary fiber, which was interesting.
Yeah. And I don't know because we haven't studied those particular short chain fatty acids to what extent they wind up at the same gene targets
Yeah,
as the other ones. I don't know. This is really just the tip of the iceberg
yeah,
because there are a lot of these modifications and we have not pursued them all.
mhm,
But I think, for the first time, we'd like to think we've made some links
yeah,
between these short chain fatty acids and some
yeah,
pretty key genes that are important for our gut health.
yeah. But sort of in the abstract here, what you told us from these results, is you eat stuff, whatever that is. That stuff gets turned into other stuff, and then ultimately, some of that stuff, in this case, the short chain fatty acids, they literally go into the cells and affect which genes get switched on and switched off, which is going to determine the proteins that are present in that cell, which is going to determine basically the health of that cell and what it's doing.
Yeah, and I don't think it's just your gut either, because
Yeah,
these short chain fatty acids, you may know, they permeate your body.
yeah,
They'll go into your heart, they'll go into immune cells. So this is probably more systemic than just your gut. But we've kind of worked out at least some of the specific mechanisms for the gut, and I think it's going to be valuable to, map that out for the rest of the body as well. How do these things help your heart, for example?
yeah, yeah. And while we're on the gut, I want to get into some of the other results from that paper that you guys did. I believe there's a connection to colon cancer here. And what did you find there?
Yeah, we also looked at cancer cells, and it's true that some of the targets are these known oncogenes and things. And so, how that is affecting cancer, and there's a known to promote cell proliferation. So, exactly how that part is working, I'm not as sure about, but we do know that they do change the gene expression and the targets do change in cancer cells. And to what extent then the fiber actually is making things worse in cancer, I don't know. But certainly for preventing cancer, I think we have pretty solid models that you're keeping good gut intestine, integrity, and I think that's valuable. For the cancer side, it's definitely true you're hitting another set of targets.
Mhm.
Some of them are involved in cell proliferation, which is what cancer is all about, as you know. If you start growing uncontrollably, you get cancer.
Mhm. And so some of the genes that these short chain fiber derived short chain fatty acids in this case affected had to do with the junctions between the cells of your intestines. So how they affect how tightly those cells are packed together, and therefore whether or not you're going to have a leaky gut and things like that.
Mhm.
Any other types of genes that were families of genes that were affected?
Yeah,
Any
there were
patterns there?
ion channels and things that are again important for gut function. You need
Yeah.
bring your ions in, from the inside to the outside. So there were quite a few different systems there, but those were some of the most obvious ones.
Mhm.
The cell integrity and the transport genes were there were quite a few of those that we
Yeah.
saw that were targets.
And remind me, in this paper, you were looking at, were you just looking at rodents, or were you looking at human cells in vitro as
We
well?
did both.
Okay.
We did it initially in human cells in vitro, and that told us what the targets were. It's very easy to map them out. And we also did human cancer cells in
Mhm.
vitro, and then we followed it up with the we basically had high fiber diets for mice and showed similar kinds of targets in the gut in the mouse. Were presumably hit by these short chain fatty acids. Not presumably, they were. So the bottom line is that that's nicer because it's a more controlled system, and we could show then that it was going on in a living organism.
Yeah, yeah. So at least directionally similar effects in the cultured human cells and in the mice.
Correct. Yeah.
The other thing, among many things that you work on, The other stuff that I thought was interesting is this concept of the lipidome and looking how the fatty acid profiles of the body change in health, in disease, in aging. Can you start to talk about that a little bit? Give us a sense for what the lipidome is and how big and how diverse it is.
Well, it's huge. I mean, we don't even know all the lipid molecules out there because you can get many, many permutations of lipids. So lipids, people just thinking, oh, they're greasy molecules. But they've always had kind of a bad reputation, I guess, because of that connotation. But they're critical for your cell integrity. They make up your membranes are lipids, and obviously there are just tons of those. But it turns out they're very important signaling molecules too, sphingolipids, ceramides, things like this. And it's an almost infinite number of molecules, it seems, because you can get all these different permutations depending. Again, this gets into chemistry, but whether the bonds they're called saturated versus unsaturated, which,
Yeah.
by the way, is a big deal. But anyway, so we don't know what most lipids do beyond, the integrity stuff in the cell, and especially how they signal. So we just started profiling them. We know they're important for energy metabolism. For example, when you run, yes, you use up your glucose right away, but then you start using your fatty acids for
Mhm.
metabolic purposes, carnitines and things like this that get derived from these. So anyway, so we just started profiling them through health and disease just to see what shifts are there. And so we're getting some initial observations about, again, what some of these things might do. So we started associated some classes of lipids, for example, as pro-inflammatory, others as anti-inflammatory, in addition to ones that are already known. There's some characterized things out there already known to be important for inflammation, for example. But we found new sets that I think are going to be useful to know about. No, they could wind up being important markers of disease then.
Yeah, yeah. I believe in that paper you looked at things like the transition into insulin resistance and other disordered states. Can you talk about that a little bit? How some of these, going from a state of health to unhealth or vice versa. How did some of those transitions look in terms of how the lipid profile changed?
Yeah, I mean, it's pretty complex. I would say it's not just lipids. We'll see whole metabolic profile shift as well. So that particular paper, I think, focused on lipids, but we have others with a little more focus on general metabolites as a whole. And so I guess I can say that there are classes of molecules that shift between, as you become insulin resistant. And we're trying to see if we can get predictive of this early, meaning if your baseline profile could actually predict whether you'll get there. We're not there yet,
Mhm.
but that would be a very nice thing to figure out because then you can try and maybe modify coming back to this diet idea. And this is one reason why we like studying the lipidome and the metabolome as you call these things, is because we hope to be able to ultimately modify people's diet to just keep them in a healthy state for a longer period of time. So on the classes of lipids that changed, yeah, I mean, it's pretty clear the long chain fatty acids and short chain, their balance is a big deal.
Mhm.
Short chains are used a lot for energy, although long chain can in certain circumstances, we think. So, yeah, I guess I'm not sure what to say beyond that. I think there's still a lot to figure out, but
Yeah. Now, so I would imagine, and I know there's people, different people out there who work on this. To what extent have you worked on assessing the extent to which changing dietary fatty acid composition leads to changes in the lipidome? I mean, inevitably there's going to be some change there. Is dietary fatty acid composition a very strong driver of what the lipidome profile of a person will actually look like?
this is a great question. I don't know that as much as I probably should, to be honest. But I do know that it's not a one-to-one correlation.
Mhm.
And there's a very interesting article out there or study out there where someone ate 720 eggs, ate nothing but eggs for a month. And the idea was for most people, look, if you eat that many eggs, your cholesterol is going to skyrocket, right? And it turns out his went down.
Hmm.
Now,
This was a person who was well, yeah, unpack it for us a little bit more.
yeah. So this is a person, he's kind of a thin, hyper metabolizing kind of guy. But it comes back to this idea about personalized metabolic states.
Yeah.
So in his case, his cholesterol, it's not excessively high to begin with. And it's thought he liked to think that because of his particular exercise profile, he would not be likely to get high cholesterol. So he tested this by eating literally nothing but eggs for a month,
Hmm.
720 of them. And, the textbook would tell you, you eat eggs like that, you're going to skyrocket. And that's probably true for the average person. But there's sets of people who, again, your liver does a lot of converting of lipids and fats into glycogen and carbs. So you have we are homeostatic systems with a balance between carbohydrate and lipid metabolism, and we're all a bit different.
Yeah.
And so what one person eats for lipids may not necessarily turn into high
Yeah.
cholesterol in their
Yeah.
blood. Does that
Well, I
make
mean,
sense?
yeah, a very important principle for people to understand is that, the particulars of your diet are always going to be context dependent. So just so people are super clear, we're talking about Nick Norwitz, who I've talked to previously on the podcast. If people want to go listen to
Oh,
that.
great.
But so at baseline, he's not like the average American, right? He is what they call a lean mass hyperresponder. He eats a high fat ketogenic diet. So his baseline metabolism, baseline diet are already quite different from the average person out there.
Right.
And I guess, in the abstract, the takeaway here is what you eat and how you eat it is going to depend very heavily on the larger dietary metabolic context.
And your baseline state, as you put
Yes.
it. 100% agree. Yeah. So I didn't realize you had covered all that previously. So
Yeah.
anyway,
But most people listening won't have heard that one. It's a
it's a
fun
very
story.
fascinating set. But I think the important lesson out of all this, which I think we're both getting at, is you really have to know yourself.
Yeah.
And if you are going to make a drastic change like that, you probably better follow it pretty carefully, because you don't want to put yourself, if you are out of balance, you don't want to put yourself further out of balance. And what puts you out of balance may be different from what puts me out of balance.
Yeah. I want to ask you, I mean, with that in mind, I want to go back to the diabetes question. So you
Yeah.
told us your own personal story. You told us that there are many different subtypes of diabetes, both subtypes of type 1 and type 2. Type 2 is more common, and it's what, I know a number of people with type 2 diabetes, but I don't know what their subtype is, and I don't think they do either. So if you're not Michael Snyder and you can't go into the lab and do all this deep profiling that you can do, let's say I'm the average person, middle age with type 2 diabetes. All I really know is that I have type 2 diabetes and that my doctor has said they recommend taking metformin or this or that. How would I get subtyping of my diabetes done? Is there a test I can take to know specifically if I've got the type that Michael Snyder has, or I've got another type?
Stay tuned, because we have another paper that just came out that's pretty cool that I hope to translate, which is that it turns out, so if any of you have worn a glucose monitor, you will see your curve. And what's interesting is that the shape of these curves are very different from one person to the next. And we think that represents the subtype.
Mm,
We just had this paper out where we had people drink. They call it a glucola. It's a sugar drink.
yeah.
And you can buy it. And if you're wearing one of these glucose monitors, which you can now get over the counter, from the shape of the curve, we can get pretty predictive of insulin resistance, muscle insulin resistance, and a beta cell defect with 0.9 accuracy, if
Wow.
you will.
So what you're saying is, if if you say you lined up 10 people and you gave them 10 boluses of food, the exact same food, whether it's a sugary drink or rice or whatever, their blood sugar is going to spike to some extent in each case. But everyone is going to spike a little bit different. Some will spike more, some will spike for longer or shorter periods of time. The shape of that glucose response curve.
Yeah, and some will go up very quickly and
Yeah.
down slowly, and others will go up slowly and down quickly. And again, if you think about it, it's how you're metabolizing your glucose.
Yeah.
And that reflects your underlying defect that's going on in you. So, and some people will get multiple humps in there.
Yeah. Wow.
In their curves. So and we use, AI machine learning to be able to extract features from those curves and build these predictive models. And so we started by looking at these 4 subtypes: muscle insulin resistance, this beta cell defect. It's called hepatic insulin resistance. So liver is.
Yeah.
So this is why this is a big deal, because coming back to this, you have many organ systems involved in glucose metabolism. You have muscle, you have liver, you have your brain is one of your biggest energy consumer. Pancreas, of course, all these things, and there's all these different biochemical pathways. It's a complex process. So just calling somebody a type 2, it's like looping all animals the same, calling cockroaches and humans the same. And I just don't think it's not a useful comparison.
Yeah. And right, like even insulin resistance is too broad, right? Because it's not like all of your cells are insulin resistant or insulin sensitive. It might be just your liver or mostly your liver or other organs at different times.
Yeah, we were saying liver and muscle. We know that at least 2 subtypes right there. So anyway, we're not there yet. Very good for predicting the incretin defect. That's this hormone that, again, the Ozempic equivalent, if you will, or hepatic insulin resistance. But we're quite good actually now at the muscle insulin resistance and beta cell, and muscle insulin resistance one of the most common. So soon, it's not a clinical test yet, but I hope to get this out as a home test. Back to this home testing, where you could just go to CVS, get it over the counter, glucose monitor, pick up a glucose drink at the same time from the shape of the curve, upload it into an algorithm, and then it'll say, you've got muscle insulin resistance.
Yeah.
Why is that valuable? Well, it does tell you, some of the drugs you might take, but it turns out we built other predictive models. If you're muscle insulin resistant, a lot more activity and strength training will help you more than if you have a beta cell defect.
I see. So like in your case, you said weight training didn't help. This test would have told you that ahead of time.
Correct. And I didn't realize that until after the fact. And now it all makes sense to me why this increase in muscle mass, which we quantified, it's 10 pounds. And
so what we're on the cusp of, based on your work, is you could use a just over-the-counter, regular old glucose monitor that would connect to some kind of app, say, and based on how you're responding to a sugar drink or a given food input, the shape of that curve is going to indicate with pretty good accuracy what type of insulin resistance you might have, and that's going to have very practical. It's going to have very practical recommendations as to how you would treat that through a lifestyle change.
We believe so. Yeah, you would not be, for example, a potato eater.
Yeah.
You might eat something else that wouldn't spike you as much. So that's the idea. We'll match lifestyle to the subtype you are. That's where we're going. And I'm not saying we're going to totally prevent diabetes, because age is the number 1 risk factor for diabetes. But even if you push it off for 10 years, we think that's a pretty good thing. And maybe you will ultimately still go on drugs, although maybe some of the drugs of the future will be foods, right? That are mimicking drugs, perhaps. And also they're orthogonal as well, because I am on, I mentioned before, Mounjaro, this GLP-1 agonist, but I also, I'm pretty careful about my diet too, which I do think is pretty helpful.
Yeah. Well, speaking of aging, I want to talk a little bit about the connection between metabolic health and aging. You've done some work in this realm. What can you start to tell us about sort of the speed of aging and how linear it is, because in general the average person more or less just thinks that, right, you just sort of tick along at a constant rate, and aging is this linear gradual process, but that's not necessarily the case, it seems.
Yeah, we had another study out just last year that got exactly that. So because we're profiling people over time longitudinally, we can see how they're changing. I think the most important concept was that everybody changes differently.
Hmm.
So for some people, the top biochemical pathway that was changing was their cardiac hypertrophic signaling pathway, their cardiac pathway, if you will. They were a heart ager. And for other people, like me, I'm a pretty typical ager. My metabolic and my immune pathway change. But luckily, my immune one is rather slow. But my metabolic pathway, maybe because of my dysfunction, is changing with age. But everybody's aging differently. Some people are kidney, some are liver, and so that we think that's important because if you know how you're aging, you can adjust your lifestyle specifically in those areas. Comes back to what I was saying before about the metabolic testing. But then the other thing we just asked was exactly the point you just raised, that everybody thinks that, of aging as a gradual process, and it didn't fully make sense to us. So we just looked at it. We like to let the data tell us what's going on. And sure enough, these different pathways change at different times. And then we simply ask, well, are there certain times in people's life where you see the biggest changes occurring? And we saw that there were two births. One is in the 60s, which we expected. It's well known that as you hit your 60s, disease risk goes up for virtually all diseases. Your immune system declines, and that's why you get vaccinated. It's pretty important to get flu vaccines and COVID vaccines when you're in your 60s because we saw that. You just saw your viral response pathways are definitely diminished. And then we saw other things that weren't necessarily known. Kidney function dropped, carbohydrate a bit. Muscle mass we saw, but that was expected. And skin, you can visibly see some of those things.
And how are you actually assessing this? How did you make these measurements?
Yeah, it's mostly out of plasma markers. So we'll see certain proteins. So, for example, your extracellular matrix proteins, which would be in the skin and in the muscle, we'll see those shifting in your blood relative to earlier times. And we'll see big changes as people start hitting their 60s. So your blood is, as you know, it's a pretty powerful a way of monitoring people's health because it runs throughout all your organs. And so it's a general, by profiling blood, we hope to get a picture of people's health. Mm-hmm. And so we do see all these biochemical changes. The other big time of change, though, that we saw was this one in the 40s. We said 44, but it's fairly, that's just where it peaks. It's kind of broad. And there, that wasn't necessarily expected. Same thing, we saw muscle protein shifting. We saw lipids shifting. We saw caffeine and alcohol shifting. So, which I thought was very interesting. And anecdotally, no sooner did we publish than I got all kinds of emails about this stuff from people said, boy, that makes a lot of sense. When I hit my 40s, I pulled my muscles and all this stuff. And I think it does make sense. We're going to follow this up, but people's fat deposition shifts when they hit their 40s, for example. And I think that fits with these lipid changes we see. And I think this is a big deal from actionability once again, because I think, as you hit your 40s, maybe you better keep a little bit closer eye on some of this stuff because, and lipids being a good example, if you start going off, statins are pretty safe. I'm on statins. It's probably a good thing to do. And some of the other things, like alcohol metabolism, is true for me, and caffeine metabolism especially. I don't drink. I had to stop drinking coffee when I hit my 40s afternoon, because it would keep me up all night.
I see. I see. So you're probably not metabolizing it as well or as quickly.
Yeah, that's the thought on that one. Sometimes we can't say directionality of these things. We just see the changes. And so I think for both of those, the alcohol and caffeine, we saw shifts. And the muscle one probably makes sense. Now, the mechanism why that's the case, what's going on here isn't so clear. Our hypothesis is that, people are pretty active in their 30 and in their 20s and such. As they hit their 30s, they tend to slow down a little bit 40. So the hypothesis is that we're not exercising enough and probably not eating as well as we should. And so it catches up with you when you hit your 40s. That's our hypothesis. So we're trying to look at people who have maintained a healthier lifestyle to see, if they're able to push those changes off to a later period.
Yeah. And so I guess one of the key results here is that you saw this sort of nonlinear burstiness to the speed of aging in essence. It wasn't this gradual progressive thing. It was progressive. It obviously people just do age over time, but you had these kind of bursts.
You'll see, some things you'll just see change over the whole time. Yeah.
You on average you saw these kind of bursts in roughly the 40s and then again in the 60s. So it sounds like you don't quite know what accounts for that yet. You think it could be something, there could just be kind of a systematic correlation in lifestyle factors where people are slowing down or changing
That's our thought for the 40s. I think the 60s is, it probably is your immune system is at least one of the culprits and that's integrated with every condition you can think of. So, cancer goes up as you hit you hit your 60s, and your immune system is absolutely critical for clearing your cancer. We're getting cancer our whole life, and your immune system's clearing it. And so the general prevailing view is that as your immune system declines, the chances of some of the stuff escaping goes up, and therefore you get full-blown cancer as you hit your 60s.
Yeah. So what are, towards the beginning of the conversation, you held up your hand and showed us a lot of the stuff you're wearing. Can you walk us through the stuff that you use that is available to people today? Not everyone can go in and get all of the deep sequencing stuff done from their blood, but you can get pretty comprehensive blood work done from the comfort of your own home, multiple times a year nowadays. What are some of the things that people have access to today, ordinary people, that are the most consequential and the most bang for their buck?
I'll start with the glucose monitors, continuous glucose monitors, because I've never seen anyone use a CGM, as they're called, and not change their lifestyle.
Yeah.
It's so visual that when you see, and a lot of this is obvious in hindsight. In fact, one of my favorite stories is after we published our first continuous glucose monitoring paper, the New York Times reporter called me up and he said, I thought I was eating the healthiest lunch every day. I had salmon on salad. What could be healthier than that? And then he puts on his glucose monitor and he spikes through the roof. And you can probably guess what it was due to.
Salmon and salad?
Yeah.
Was he putting some kind of dressing on it?
Bingo.
Oh, okay. There you go. Yeah.
So, and he didn't, obvious in hindsight. And so all he had to do was leave that off and he got the healthy lunch he was looking for.
Yeah, a lot of those dressings.
For me, it was a pulled pork sandwich. I wasn't officially diagnosed as diabetic as a managed diabetic. I was in that in-between phase where I'd first been diagnosed, got under control, and I had to put a glucose monitor on. And I ate this pulled pork sandwich and I just saw my glucose go up over 320. It was so right. And I showed it to this friend of mine. He goes, Mike, everybody knows pulled pork has sugar in it. Well, I didn't know, but in hindsight, it makes a lot of sense, right? And there's just a zillion cases like that. When you wear one of these glucose monitors, sometimes it's very obvious in hindsight, duh, I should have realized that. Now I'm hypersensitive to sugar. So,
yeah. I mean, I did this myself once. I once wore one. I'm not diabetic or anything. I just did it just to know. And it's similar to some of the examples you gave. At that time of my life, a few years ago, I would commonly have breakfast cereal, and I bought the quote unquote healthy cereal, whole grain, organic, vitamin fortified, blah blah blah. And that was like the only thing that was really, really, really spiking my sugar. But boy, was it a big noticeable spike.
Yeah. And then sometimes you go back and look at the package and you see they actually
high
have
fructose corn syrup
in there. And sometimes it's natural sugar, like grapes will spike you. And that's one with a very, very rapid rise. And by the way, back to one of your earlier comments, from a potato to grape ratio, we can predict your insulin resistance.
Really?
Yeah, because grapes are like sugar.
Yeah. Grapes are like, it's sort of like eating glucose, whereas potatoes is
It
starch.
is. But
Yeah.
luckily the spike is pretty transient with
Yeah.
grapes, thank goodness. But mixed berries, believe it or not, they have more fiber, we think, so they actually don't spike you as much.
Yeah. Yeah, but I mean, it's amazing what you can see just by wearing one for even just a few days. Like, I gave the example of the breakfast cereal from my own life, but I didn't spike much from starchier foods, and some
Yeah,
people do.
great for you.
Yeah.
Yeah. So that's number one. And given the, pre-diabetic, excuse me, and diabetes endemic going on, this is so important. I think everybody should wear one, ideally once a year. And then smartwatches, I think, are just no-brainers too. And you may or may not know, we actually discovered early on. So we got involved with these back when they were just fitness trackers, and we put them on our cohort. And right away, we discovered you can tell when somebody's getting ill from an infectious disease just from a smartwatch because your heart rate jumps up. And
Hmm.
it's how I first discovered my Lyme disease because my heart rate jumped up in advance of symptoms.
Really? And what
Yeah,
were your symptoms and what was the lead time there?
the symptoms appeared. So I was actually flying to Norway, and you may or may not know, but your blood oxygen drops on airplanes
Mm.
because they don't pressurize the cabin fully. It's why you get tired on airplanes.
Hmm.
And in my case, because I measure myself all the time, I saw my blood dropped abnormally low and my heart rate went up. Your heart rate will go up too, but it went up, both went up to way, way more than usual. And when I landed, they didn't come back to normal. So I knew something was up. And then the next day, I got a little bit of fever, kind of got fever off and on. Now, 2 weeks earlier, I'd been in rural Massachusetts where 55% of ticks were Lyme infested.
Hmm.
So I thought it might be Lyme. So I went to a physician there and he saw my immune cells or my monocytes. Um, and he said, yeah, you got a bacterial infection. He wanted to give me penicillin. And I said, no, I need doxycycline. I warned him ahead of time it might be Lyme because of the timing.
So he just assumed or presumed it was a bacterial issue.
Yeah, I mean, monocyte doesn't prove it, but it wasn't unreasonable to
Okay.
think that was a bacterial infection.
Okay.
It's probably what you'd normally say. And spirochetes are the Lyme spirochete is a type of bacteria. So anyway, but it got a little tense. I did tell him I wanted doxycycline. Most physicians don't like their
Why?
patients telling them. They
Oh
just
no, they don't like
they
you
don't
telling
like
them what to do.
patients
Yeah.
saying, yeah, here I am saying,
Why
well,
were you asking for doxycycline?
because I thought it might be Lyme because of this two
Okay.
week.
And that one is known to be good for Lyme.
Yeah, that's what you take for Lyme. It's much,
Okay.
much more effective. So he did give it to me, and my fever went away right away. Well, I don't even remember if it says bouncing off and on, and it cleared up right away, and I could see my heart rate went right back to normal. And when I got back, I got tested as Lyme positive. I had antibodies, even had an antigen. And because I measure myself a lot, I give him blood 3 days before I went as negative. So I seroconverted during that time.
Wow.
It's a really well-controlled experiment. So the bottom line is that yeah, I first could tell when I got Lyme from a simple, at that time I was using pulse ox. Now you get your blood oxygen from a watch. But anyway, so and my heart rate from the watch. And so we went on to retrospectively show that every time I saw my heart rate jumped up 4 times for over a 2 year period. One was a Lyme time, two were viral infections, and a fourth, I was asymptomatic, but I had high CRP, C-reactive protein, it's
Yep.
called. And every single time I saw my heart rate jump up in advance of that.
Days in advance?
Yeah. And well, more like about 30 hours. It depended on the
Okay.
particular time.
Roughly a day or so.
Yeah, roughly a day. But we wrote algorithms in to detect this. And again, so you can see, and then of course that we published that in 2017. And then along came COVID in spring 2020 is when it hit here. And right away we partnered with Fitbit and showed with 32 people who were wearing a Fitbit who had COVID, we could see that their heart rate, and they had symptom date and the diagnosis date. And we could see that for 80% of them, we could tell when they they got COVID, and we could pick it up in advance of symptoms. And there, the median is 4 days, because COVID has a long pre-symptomatic period.
That's probably
So
why it's so part of why it's so hard to control the spread.
it is. I'm sure it spreads. And in fact, the very first case, if I were to show you this, was someone whose heart rate jumped up 9 days in advance of symptoms. So we
Wow.
assumed they were running around for 9 days with this thing. And then we wrote a real-time detection. So if you're wearing a smartwatch, I'd love you to sign up, and you'd be able to. We want to perfect the algorithms, but basically, we can tell when somebody's getting COVID in advance of symptoms. Here, the median's 3 days, because it's a alerting prospective system. So we have to be sure it's up high enough before we send an alert. Anyway, 80% of the time, we can tell when you're getting COVID in advance of symptoms. And it's more sensitive than an antigen test, because we think you need as little as 2 beats per minute, and we can pick that up.
Wow.
Now, it's not specific for COVID. So any infection will
Yeah.
do it.
Yeah.
But and it also turns out that certain things like long airline flights will trigger these red alerts, and mental health is actually a trigger as well. So this is why we want to bring in more data, because I know if we bring in respiration and things, we'll tell the difference between a respiratory viral infection and mental health stress. But the bottom line, it's a general stressor. And so I think if you're just sitting around, as I like to say, listening to boring Mike Snyder, and you've got, you get a red alert, you're either mentally or physically stressed, and so something's going on. And I think in both cases that's actionable. So we're very keen on using wearables to better, detect things early, and I think this can be a very, very useful thing. Right now, we're trying this stuff for cancer and other areas as well, to see what else we can tease out of a smartwatch.
Yeah.
And so these, going back to your way earlier comment, these are simple devices and you don't need a fancy smartwatch to catch this 2 beat per minute increase to tell you whether you're getting ill or not. It's pretty obvious. And so I think those are going to spread pretty wildly, I hope. And if it's not a Ring is fine too, by the way.
Is the Ring also measuring your heart rate?
It does. They're not quite as high resolution, but the Ring is better. I'm using an Oura Ring. I'm not conflicted on that one. They're better for sleep duration and some of the sleep measurements than a watch is. They're not generally quite as high resolution, but
so the heart rate monitor is better on the watches.
I think so, yeah. And they vary from device to device. So some of the watches they're all quite good for resting heart rate and for heart rate variability, we think. And heart rate variability, for those who don't know, that's an important metric of health these days. It's becoming more and more realized that this is quite powerful. But when you run to max, some of the watches pick that up pretty well, and some aren't quite so accurate. It depends on the resolution they're set at. And some are pretty accurate for skin temperature, some not so accurate. So they vary a bit from device to the device. Some of them measure things that you don't get in a doctor's office, like galvanic stress response. That's conductance of your skin. You might say, well, why is that important? Well, it has to do with dryness. And that can either be it could reflect anemia, it can reflect diabetes. So it turns out, and in fact, in hindsight, I should have figured out when I was getting diabetic, because my skin got itchy, it gets dry. And so you can pick this kind of stuff up with a smartwatch now. You couldn't way back when, but you can now.
In your opinion, based on your use and seeing the data and stuff, what are some of the best smartwatches out there in terms of their sensitivity and how good and accurate the measurements are?
Yeah, it's a good question, and I'll have to watch it. Of course, here I'm conflicted, and I have a company called Sensomics, so
Yeah.
we build it for health monitoring, and I haven't tested them all, but a lot of people say Whoop is good, but I personally haven't done that because I'm too cheap to pay $30 a month kind of thing. But here's a style factor, like Fitbit and Garmin. They charge fast. The charge lasts 5 days. The number one problem with monitoring from a watch is that people don't keep the watch charged all the time. So the Apple is probably a better watch. It's higher resolution, but you miss a lot of people miss a lot of data because you can't charge it in 20 minutes. So I charge mine when I'm in the shower, but
Ah.
for Apple Watch, you're going to have to give it a little extra boost somewhere.
Oh, so you use the Apple Watch, but it takes
Yeah,
longer
I
to
have an
charge it.
yeah, so I have an Apple Ultra and I have a Fitbit on this arm. I normally have a Garmin and my
Yeah.
company sends some here.
Why are you wearing both the Fitbit and the Apple?
Because I like to compare all these things. So, for
Ah,
example, when I got COVID, I was wearing four watches, and the goal was to see which one would be the best and
yeah,
why. Like
yeah,
which parameters and what resolution is
yeah.
optimal for picking
What was
stuff
the
up.
answer?
Well, I don't have it fully up, but it didn't help that I ran out of data on one of them because the watch wasn't charged. But back to my point. Uh, yeah, the Fitbit did well. It actually, again, picked up when I got COVID before. We're still analyzing the data. Should have, got too busy to probably pin it all down. But they all work. The question is which works the best so we can tune the parameters optimally. Because I don't care about reading email and answering my phone on the watch. For me, that's
Yes.
irrelevant.
Yeah.
I want to do health monitoring. So I want to tune a watch to be as precise as possible for following my health.
Yeah.
And ultimately follow more than what we've been talking about, the
Yeah.
just general, physiological state, which I hope will pick up
Yeah.
cancer, heart conditions, other things.
Yeah. So in terms of what are ones that measure heart rate and the galvanic response that don't necessarily have all the bells and whistles of the Apple Watch, like email and stuff? If you just want to do heart monitoring, which ones are good enough at detecting things?
Even a Fitbit, even
Sounds like Fitbit works.
yeah, and again, because people it's easy for them to keep it charged, and they're $150 these days, maybe $160 for a pretty good one, charge six. Uh, so even my daughter's wearing one now. I didn't.
It's called a Charge 6.
Yeah, and there's another one that's probably has more bells and whistles if you want. Anyway,
What is it?
that's the Sense 2, I think, is their latest one. They may even have another one out now. That's pretty good too. And we'll see what happens because you may know there's a Google Watch out there now, and Google owns Fitbit. So we'll see how that all evolves out of this. And but my understanding is that the Pixel will become for health monitoring from Google, and the Fitbit will be more for the tracking sorts of just the fitness sorts of things. But we'll see what happens. Again, that's not. I'm not conflicted on that one.
And you are working on a watch, or it's already out, or
Yeah, so I have it doesn't go direct to consumer. You'd have to be in one of our research studies, and it's built for health monitoring. It's called Sensomics.
yeah,
Yeah, so if you get in one of our studies that's using it, you could get one of these. We just don't have you need a big technical support team if you're going to go direct to consumer.
yeah,
And so we will go there, but right now we're just rolling out as research, and
yeah.
it's meant to be optimal for health monitoring, trying to get the resolution. It even does blood pressure. So
We've already covered a lot, touched on a lot of different stuff. Is there anything you want to reiterate, or anything you feel like we didn't unpack fully for people that you want to talk about?
we've covered a lot, as you say. I guess, one thing people often ask about is privacy. It's not something I worry about because I have 2 petabytes of data you can download right now. Nobody's ever abused me on it. So I personally think the benefits of this kind of health monitoring and such greatly outweigh any concerns about privacy concerns. I think it's really important for people to monitor their health while they're healthy. I guess I probably hammered this already through this podcast, but the takeaway lesson is just that. I think you really should know your baseline, really know your health state, and there's a tendency to push things off, wait until you get ill. But I'll give you a good example, like whole body MRIs, which again, this company QBio I'm involved in is doing. I think they're so important because if everybody has nodules, if you ask any physician today, should you do a whole body MRI? 100% of the time they say, absolutely not. And they're worried that you're going to find nodules. And if you
Meaning
ask,
like benign things that don't matter.
Exactly. And if you ask me, of course, I say absolutely you should. And the issue isn't do you have nodules? Everybody has them. Men have them if you go high enough resolution. Men have them in their prostate, women in their ovaries, 100% of the time. But the issue isn't do you have nodules? The issue is do you have any growing nodules?
Right.
The only way you know that's through the longitudinal.
I'm serious.
So I think that's a big lesson to get out of this too, that really get your healthy baseline and do it periodically so that you can see what's going on because these trajectories, I think, are the most important thing.
Yeah.
And a good example I'll give you on the MRI was a friend of mine, had cancer on his spine, woke up one morning, couldn't move his arm. Then they, took him in, got an MRI on the spine, saw this nodule, cut it out, fixed everything, then went back a month later and did his whole body MRI, and they found 3 more nodules on his spine. So were they there to begin with, or had it metastasized? No way to know.
Yeah.
Never had a baseline.
Yeah, but you needed that double
You need
snapshot to
that.
see it.
Yeah. So in my case, I have 9 nodules. I've had 19 whole body MRIs over the last 8 years. And I have 9 nodules. I'm happy to say none are growing. But if I ever get cancer, I know where they are and I know what their size is. So I think this is a big deal. Again, this concept of healthy baseline, I think everyone should get it.
And how much does an MRI cost someone if they just go in and want to get one just for the hell of it?
Yeah, I think pre-novo, you have to look it up, but it's on the order of like a little over $2,000. I think there's some cheaper ones now. How good they are, I can't attest. My company, Cube Bio, they charge $3,500, but they also do a very deep molecular profile and such as well. And back to this point you said before about what do you do with all the data? They display it back in a fashion you can understand, not just your physician, that you can look at this and say, well, these things look good. These areas I could use to do better on.
Yeah. All right. Well, that's fascinating stuff. A lot of useful stuff on the wearables and the tech here in this conversation. So I want to thank you again, Professor Snyder, for your time. I really appreciate it. Any final words you want to leave people with?
Stay healthy. I think we all can. I thought one more thing. Can I add that?
Yeah.
So the difference between, and this is really, again, a big driver for us, the difference between people's health span and their lifespan is 11 to 15 years, depends how you measure it.
On average.
On average. So that means people are living, on average, the last decade of their life unhealthy. And we've got to fix that. So that's the message I want to leave people with. Let's all live long, healthy lives, because that's what you want to do. You want to live healthy and then just, blah, blah. So that's how we should be.
All right. Professor Snyder, thank you for your time.
Thanks for having me.
Hey, everyone. I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.